Hello everyone, I am Dr. Vamangi Goswami. Today I would like to discuss about disaccharides, in which we will study the structure of sucrose, maltose, cellobiose and lactose in details. This topic belongs to Gujarat University, B.Sc Chemistry Semester 5, Paper 301, Unit number 4A. So let's begin. Now first of all, let us understand what are disaccharides. Disaccharides are simplest and most important oligosaccharides. In general, they are sweet in taste, crystalline and water soluble substances. They are easily hydrolyzed by enzyme and dilute mineral acids to two monosaccharide units. These arise formally by intermolecular dehydration between two monosaccharide molecules. Disaccharides on hydrolysis gives two monosaccharides units which may be same or different. Those two are joined together by glycoside linkage. Now we have to study four different types of disaccharides in detail. First is sucrose, maltose, cellobiose and lactose. Sucrose is formed of two different monosaccharides. One is alpha D glucose and beta D fructose, while maltose is made up of alpha D glucose and alpha or beta D glucose. Cellobiose is made up of beta D glucose and alpha and or beta D glucose, while lactose is made up of beta D galactose and alpha or beta D glucose. Now let us start with our first disaccharide, sucrose. Sucrose is the commonest sugar known and familiar disaccharide. It is also known as table sugar. The most important sources are sugar cane and sugar beet. It is also present in varying concentrations in maple saps, honey, fruits, flowers, seeds and roots. On hydrolysis with acids or enzyme invertase, sucrose gives two equal parts of D-glucose and D-fructose, which thus constitute the two monosaccharide units of sucrose. From combustion analysis and molecular weight determination data, the molecular formula of sucrose can be determined as C12H22O11. Sucrose neither reacts with phenylhydrazine to give oxazole nor reduce Fehling solution or tolerance reagent, indicating that the carbonyl group of both the monosaccharide are involved in linkage, that is glucose is linked to its C1 to the C2 of fructose, that we will see later on. So thus it is indicating that sucrose is non-reducing sugar. Now let us see the structural part of sucrose. As we have seen, sucrose is made up of two monosaccharide units, that is alpha D glucose, that is alpha D glucopyranose, and beta D fructose, that is beta D fructofuranose. Now star shows the anomeric carbon of both the monosaccharides. Let us first number the carbon atoms. Carbon number 1 in glucose, carbon number 2, carbon number 3, carbon number 4, carbon number 5 and carbon number 6. Here carbon number 1 or glucose is anomeric, while here in beta D fructofuranose let us do the numbering first, 1, 2, 3, 4, 5 and 6, right. So carbon number 2 is anomeric over here. So here anomeric carbon of both the monosaccharides are leaving one H plus and one OH minus and leaves one water molecule, releases one water molecules and forms one glycosidic linkage between the two monosaccharide units. Those forms the structure of sucrose. Now let us understand the naming or nomenclature of the disaccharide. Disaccharide is formed of two monosaccharide units. First monosaccharide unit, the suffix is added syl sil, while the second monosaccharide unit, the suffix added side. So the name of this particular disaccharide is alpha D glucopyranosyl beta D fructofuranose. Sucrose shows one important phenomenon known as invert sugar. The hydrolysis is accompanied by change in the sign of rotation from positive to negative. It is often also called the inversion of sugar. The levorotatory mixture of D-glucose, D plus glucose and D minus fructose obtained has been called invert sugar, while here plus sucrose has specific rotation of plus 66.5 degrees and plus D plus glucose has specific rotation of plus 52.7 degrees, while D minus fructose has a large negative specific rotation of minus 92.4 degrees, giving a net negative value for the whole specific rotation of the mixture. Now let us start with our second disaccharide, maltose. It is obtained by partial hydrolysis of starch. It can be produced by microbes in fermentation of starch to ethanol. On hydrolysis in presence of enzyme amylase or diastase that is found in malt, that is sprouted barley, on hydrolysis of 1 mole of maltose gives 2 moles of D-glucose, right? From combustion analysis and molecular weight determination, molecular formula of maltose is derived as C12H22O11. Now let us see the structure of maltose. As we have seen earlier, maltose is formed of two glucose unit, that is 2 alpha D glucopyranose. Here one anomeric carbon of one glucose, that is carbon number 1 of one glucose, hydroxyl group on carbon number 1 of glucose, and carbon number 4 of second glucose has another hydroxyl group. From it, H plus and OH minus will be released as water molecule and forms a glycosidic linkage between those two molecules of glucose that forms a structure of maltose. This glycosidic bond is in between carbon number 1 of glucose and carbon number 4 of another glucose. So the nomenclature of maltose is alpha D glucopyranosyl 1 to 4 linkage alpha D glucopyranoside. Now let us see some properties of maltose. It reduces tolerance reagent and Fehling solution. That is why this sugar is a reducing sugar. It also forms ozazone while reacting with phenylhydrazine. Maltose exhibit phenomenon of mutarotation as the free anomeric carbon of reducing end of maltose is more favored in beta orientation than in alpha orientation. So maltose in alpha form gives specific rotation plus 168, while in beta form it gives plus 112 degrees, while after mutarotation in the solution equilibrium, at equilibrium it gives specific rotation 136 degrees. Now let us see the structural determination of maltose. Maltose can be easily oxidized in presence of bromine water to give a monocarboxylic acid, maltobionic acid, which has an open ring structure and has one COOH group. After complete methylation of this maltobionic acid, it gives octamethyl derivative of maltobionic acid. Followed by hydrolysis, it gives us 2,3,4,6 tetra-O-methyl glucopyranose and 2,3,5,6 tetra-O-methyl gluconic acid. Here maltobionic acid after methylation gives us octamethyl derivative. So here all the free H plus ions are replaced by methyl groups. After hydrolysis it gives us two different monomers, and that is 2,3,4,6 tetramethyl glucose and 2,3,4,6 tetramethyl gluconic acid. The presence of free hydroxyl group in these products at position C1 and at position C4 respectively indicates that the glycosidic linkage in maltose involved these carbon atoms and that the original ring sizes in maltose were pyranose. So methylation of maltose followed by hydrolysis gives 2 moles of 2,3,4,6 tetramethyl glucose and 2,3,4,6 tetramethyl gluconic acid proves that C1 of glucose is attached with C4 of the glucose molecule. So by this we can prove that structure of maltose has 2 D plus glucose attached with a glycosidic linkage. So the structure of maltose is proved as alpha D glucopyranosyl, alpha D glucopyranoside. Now let us start with our next disaccharide cellobiose. When cellulose is treated with acetic anhydride and sulfuric acid for several days, cellobiose octaacetate is obtained, which on alkaline hydrolysis gives plus cellobiose. In the living system, cellobiose is obtained by enzyme cellulase treatment on cellulose, which is not found in humans but found in the gut of cow. Combustion analysis and molecular weight determination data arrives the molecular formula of cellobiose as C12H22O11. While cellobiose differs from maltose in one respect, it is hydrolyzed by enzyme emulsion and not by the maltase or diastase, giving 2 moles of glucose molecules. So the cellobiose is made up of 2 monosaccharide unit, that is 2 moles of D plus glucose. Thus the cellobiose has beta glucoside linkage present, as the maltose also has glucose plus glucose monosaccharide, but it has alpha glucoside linkages, while in cellobiose has beta glucoside linkages. Now let us see the structure of cellobiose. As we have seen earlier, the cellobiose is formed of 2 glucose units. These 2 glucose units, from which one of the glucose unit, carbon number 1, possess hydroxyl group, and carbon number 4 also possess another hydroxyl group. From that, carbon number 1 and carbon number 4 of this hydroxyl units releases one water molecule and forms glycosidic linkage in between carbon number 1 of first glucose and carbon number 4 of second glucose and forms beta glycosidic linkage. This is the structure of cellobiose, and the nomenclature of cellobiose is beta D glucopyranosyl 1,2,4 linkage beta D glucopyranoside. Now let us see the properties of cellobiose. It is a reducing sugar and can be inferred by its positive reactions with tolerance reagent and Fehling solution. It also gives ozazone by reacting with phenylhydrazine. Cellulose exhibit phenomenon of mutarotation, as free anomeric carbon of reducing end of cellobiose is more favored in beta orientation than in alpha orientation, while cellobiose in alpha form gives specific rotation plus 72 degrees, while beta form of cellobiose gives specific rotation plus 16 degree, while at equilibrium after mutarotation the specific rotation changes to plus 35 degrees. Now let us see the structural determination of cellobiose. Cellobiose, after oxidation in presence of bromine water, it forms cellobionic acid. Here the bond breaks and the open chain structure containing one COOH group is formed, which is known as cellobionic acid. Cellobionic acid, after complete methylation, it forms octamethyl derivative. All the free H plus are replaced by methyl groups, while subsequent or continuous hydrolysis of this octamethyl derivative forms two different monomers, that is 2,3,4,6 tetramethyl beta glucose and 2,3,4,6 tetramethyl gluconic acid. So all the above arguments prove that the cellobiose, both the glucose structure have pyranose structure, that is 6-membered ring containing oxygen, and the structure number 1 of glucose, that is carbon number 1 of first glucose, is attached to carbon number 4 of second glucose with the beta linkage. Beta linkage is present in the cellobiose, while alpha linkage is present in maltose. Now let us see our last disaccharide, that is lactose. Lactose is known as milk sugar because it occurs in the milk of humans, cows and other mammals. Human milk contains about 7.5% lactose and cow's milk contains about 4.5%. This sugar is one of the lowest ranking in terms of sweetness, being about 1/6 as sweet as sucrose. Lactose is produced commercially from whey, a byproduct in the manufacture of cheese. It is important as an infant food and in the production of penicillin. Lactose is a reducing sugar composed of one molecule of D-galactose and one molecule of D-glucose joined by beta-1,4 glycosidic bond. From combustion analysis and molecular weight determination data, the molecular weight of lactose arrived at C12H22O11. Now let us see the structure of lactose. As we have seen earlier, lactose is formed of two monomeric units, that is beta D galactose plus beta D glucose, that is beta D galactopyranose and beta D glucopyranose. Here hydroxyl group present on carbon number 1 in galactose and hydroxyl group present in carbon number 4 in glucose removes one water molecule and forms one glycosidic linkage in between anomeric carbon of galactose and carbon number 4 of glucose and forms beta linkage in between these two monomer, forms plus cellobiose. The nomenclature of lactose arrived at beta D galactopyranosyl 1 to 4 beta D glucopyranoside. Now let us see the properties of lactose. It is a reducing sugar and can be inferred by positive reaction with Tollens reagent and Fehling solution. It is also gives ozazone formation by reacting with phenylhydrazine. Lactose exhibit phenomenon of mutarotation also. Lactose is hydrolyzed by an enzyme lactase that yields equimolar amounts of D-galactose and D plus glucose. Now let us see the structure determination of lactose. Lactose after oxidation in presence of bromine water forms lactobionic acid. Here the bond breaks and open chain COOH group is attached. After formation of lactobionic acid, this undergoes methylation in presence of dimethyl sulfate in presence of NaOH. All the 8 H plus ion which are free are replaced by methyl ions and forms octamethyl derivative of lactobionic acid. This octamethyl derivative of lactobionic acid further subsequent hydrolysis gives us two different monomer unit, that is 2346 tetramethyl beta galactose and 2356 tetramethyl gluconic acid. So the formation of 2356 tetramethyl gluconic acid suggests that C5 of the reducing sugar is involved in the pyranose ring formation and C4 of the glucose linkage is present in lactose formation. So C4 of glucose is linked to C1 of galactose. So here C1 to C4 linkage of lactose is confirmed by this reaction.